Bismillahirrahmanirrahim. June 24, paper 4-3, video 3, question 8, question 9, 10 solved here. Bismillahirrahmanirrahim. We start question number 8. Figure 8.1 is a diagram of a mitochondrion. And you can see the labeling here. There's outer membrane and there's inner membrane. And they expect us to really revise this chapter well and give us it says outline the roles played by the mitochondrial membranes in respiration. So what are the roles played by mitochondrial membranes in respiration? Is allows the entry of oxygen for oxidative phosphorylation, pyruvate for the link reaction, allows the exit of carbon dioxide from the Krebs cycle and the link reaction. Then it compartmentalizes. I mean, there's a compartment of the inner and the between the inner and the outer membrane called the intermembrane space. Then the inner membrane has the ETC to release energy to pump protons into the intermembrane space. Then the ATP synthase is located on the inner membrane for the production of ATP. Now you can see it allows the entry of oxygen for oxidative phosphorylation, allows the exit of CO2 from the Krebs cycle and the link reaction, forms the intermembrane space as a compartment. And then if you go into more details about the inner membrane, ETC located here to release energy to pump protons into the intermembrane space, and then location of ATP synthase for ATP production. And then infoldings increase the surface area for many ATP synthases. You see, because this folded, the outer membrane is not folded, so we will have many ATP synthases here. More here, more here, more here. So there will be many ATP synthases embedded in the inner membrane. So infoldings increase the surface area for many ATP synthases. Then impermeable to protons to maintain high H plus concentration in the intermembrane space. You see, the H plus, when it collects here in the intermembrane space, now it can't go out and it can't come back in. So impermeable to protons to maintain the high H plus concentration in the intermembrane space and its site of chemiosmosis. Then coming to B part of the question, the shapes and numbers of mitochondria are continually changing due to fission. Fission occurs when one mitochondrion splits to form two mitochondria. Remember we said bacteria divide by binary fission. So this is another sort of fission. Suggest reasons why mitochondria carry out fission. So refers to what are you going to talk about? You talk about the energy needs of the cell. If the energy needs of the cell are increased, so there should be more mitochondria available. Then in cells about to undergo mitosis, the organelles replicate. Remember that AS level, G1 phase of interphase. Then we also need to replace the damaged mitochondria. So that is why we say reasons why mitochondria carry out fission. Then we come on to the part 2 of the question. Guanosine triphosphate GTP is a molecule that is used as a source of energy in some reactions instead of ATP. Guanosine triphosphate. So we had adenosine triphosphate, sorry. Now we have guanosine triphosphate. Now remember, adenine and guanine are both purines. So guanosine is composed of a purine similar to adenine and ribose. Suggest why GTP can be a suitable source of energy in some reactions. It will be easily hydrolyzed to release energy, or you can say the G protein binds to GTP rather than to ATP. Why? Because you see, the fact that ATP can be hydrolyzed, converted to ADP, and then energy is released. So the same thing can happen here, hydrolyzed or bond broken to release energy. Degraded by hydrolytic enzymes inside lysosomes, components of the breakdown used to produce new mitochondria. Question 9, A part figure 9.1 shows how the mean global atmospheric carbon dioxide concentration has changed over the 810 to the past 3 years leading up to the year 2020. So you can see the mean global atmosphere carbon dioxide concentration in milligram per meter cube and years before 2020 in 10 days to the power 3 the year 2020. Calculate the percentage increase in carbon dioxide concentration between point A, this is point A, and the year 2020. Point A and the year 2020. Show you're working your answer to one decimal place. You forget to read that. And this is only Gen Z who does that. And then you calculate the percentage increase. How do we calculate that? I'm sure you all know how to do that. It's 367%. So the percentage increase is 376%. How is it? It's 738 minus 540 over 540 into 100. Now, of course, the answer comes to be 36.66666, something like that. So you can see the answer is 36.7% because the answer was to one decimal place. So you didn't have to give me this whole thing. If you gave me this whole thing, this would be only one mark. You won't get the second mark for it. So this was 36.7%. I'm sorry, in the mark scheme, it's written wrong at 367, but it's 36.7%. Suggest how the changes in carbon dioxide concentration between A and the year 2020 may have affected the environment and biodiversity. Now, that of course is a very, it's come many times in your exams on this sort of a question. Increase in temperature means global warming, sea level increases, habitat changes, there's going to be forest fires, ocean acidification, ecosystems disrupted, changes in biodiversity, and of course, is going to all lead to extinction. So, increase in temperature means this could be global warming, and this will result in sea levels increasing, habitat change, forest fires could be resulting, or ocean acidification results. Food webs and food chains and ecosystem are disrupted. There's going to be change in biodiversity. You see, if trees die, then the birds that nest on those trees die as well. So, if there is a change in the plants, there's less grass, so less herbivorous animals or less trees, so less birds. So, or most of them, then this will of course result in extinction on a very large scale. And then we come to the B part of the question, outline reasons for maintaining plant biodiversity. What are the reasons for it? Because we may have future use of these plants. Then, of course, ethical reasons. We must leave the earth as we found it for future generations. Then some plants may have medical uses, so medicinal value, and we've lost them, then we've lost their medicinal value. Then, of course, plants where there is a lot of nature and reserves, and there's ecotourism. People visit those places, and then, the economy of the country is affected. People visit, live in hotels, buy souvenirs. So that's called ecotourism. Then, of course, it maintains the stability in the ecosystem. Plant biodiversity maintains, you see, producers, then primary consumers, then secondary consumers. Then plants are a resource material like for wood for buildings, fiber for cloth, and food for humans. Then, of course, maintaining plants would maintain the gene pool or the genetic diversity that we have. And of course, plants increase the soil stability and prevent desertification. So, plant biodiversity is all very, very important. You can't just think that, okay, plants don't matter much. They do matter. So plant biodiversity is of course very very essential because you might have the plants that we have now may have a future use because they contain genes for drought resistance, or maybe they have a very hardy stem so they can withstand a lot of wind. And if we lose these plants, then we lose their genome. Then aesthetic, ethical, cultural reasons, of course, plants look nice. I mean, they are a great inspiration for artists and poets. Then medicinal value, ecotourism, maintaining stability in ecosystems, resource material, wood for buildings, fiber for cloth, food for humans. Any one of those was fine. And soil stability and prevents desertification. Last question, question number 10. Figure 10.1 is a diagram of a part of a neuron membrane while the resting potential is maintained. On figure 10.1, use label signs related to label K, which is the potassium ions, which will be these ones, because remember what is the mnemonic for this is Nokia's 3 sodium out, 2 potassium in. So this would be the sodium and 2 potassium in, so we'd have to label, and then of course this would be the use label letters to label K potassium ions and ATP. So this would be the ATP. So well, of course you didn't have to write ATP. You just had to write here you had to write K, and here you had to write just A. So A was here and K was there. This of course would be the sodium ions. Of course you weren't supposed to label the sodium ions. But this, if you had to label the sodium ions, then it would have been these ones, or the 3 sodium out, 2 potassium in. So we're not labeling the sodium ions. Then it says describe the sequence, direct question, describe the sequence of events that occur during an action potential. And that's 4 marks. So I'm sure everybody knows that sodium channels open, sodium ions enter in, membrane depolarized, then there is a threshold potential reached, and then potassium channels open, potassium moves out, repolarization of the membrane, then there's a slight hyperpolarization of the membrane when it goes down, then return to resting potential, and entry of sodium ion causes more sodium channels to open. So sequence of events that occur during an action potential is basically the depolarization, this is the overshoot, and then normal. And this is because of sodium ions entering. That's why the mnemonic is Nike. The depolarization is because of sodium in and is because of the potassium exit. So this would be the depolarization and this would be the repolarization. And then this is the overshoot, which is the hyperpolarization. And then this would be the resting potential, which is maintained. This would be the resting potential, which is maintained here. So sodium channels open, so membrane depolarized, threshold potential reached, potassium channels open, causes repolarization, hyperpolarization occurs, return to resting potential. So all these points and any 4 of these got you your 4 out of 4. And then we come to the last part of the paper and the last part of the question. Table 10.1 shows the axon diameter, myelination and transmission speed of impulse of motor neurons for 3 animals, squid, cockroach and cat. So the axon diameter is 1.5, 0.05, 0.02, myelination, no, no, yes, and transmission speed is 30, 10, and 100. So if myelinators were present, it was 100 meters per second, no myelination was 10 meters per second and 30 meters per second, then the axon diameter was 1.5. So describe and suggest explanations for the result. So describe and suggest. So describe is very easy. Describe, you could have come up with myelinated axon produce faster transmission speed. Larger the diameter results in faster transmission speed, and myelination has a greater effect on transmission speed than axon diameter. So you can see the myelination was in this one, and this is 100 as compared to the diameter changing from 1.5 decreasing to 0.05. So it only decreased 1/3. From 30 it went down to 10. So in describe, as I always say, there's no biology and you just have to give me description. So myelination produces faster transmission. This is of course here, we're getting this out here because this is myelinated. Then larger the diameter results in faster transmission. Larger the diameter was 1.5 was 30 and 0.05 was 10. And myelination has a greater effect on transmission than axon diameter. Now the explanation of course comes in very easy. Myelination means there's going to be saltatory conduction, and which means that the action potential jumps from node to node. And axons with a wider diameter have less ion leakage, or you can say have a larger surface area. So if the diameter is more, that means larger surface area. So that is all what you have to give me for 4 marks. So if you look at the describe basically was telling you about the figures that was given to you. So describe, I always says no biology. So myelination, faster transmission speed, that's what you gathered from the table 10.1. Larger diameter results in faster transmission speed, and myelination has a greater effect on transmission speed than axon diameter. And the explanation, of course, is myelin sheath results causes when the axons have a myelin sheath, this results in saltatory conduction, and that is, of course, faster. Or you could say action potential jumps from node to node. That, in fact, is the explanation of saltatory conduction. But if you said saltatory conduction, that means you knew a biological term, so we would give you a mark for that. If somebody didn't remember it and wrote action potential jumps from node to node, well, fine, we'd give them a mark for that. But it's the same mark scheme point. So either you gave me this point or you gave me this point as one mark. So if you just wrote saltatory and then you gave the explanation as well, I wasn't going to give you 2 marks. Then axons with larger diameter have a larger surface area, or you can say they have a lower resistance. So that is why axons with large diameter have a slightly more faster transmission speed. So this ends this paper, and I hope this has been helpful to you. Please do leave comments in the comment box and let me know if there is anything which I can improve or I can help you in some way. Thank you very much.